Good day, everyone. This experiment is called Membrane Transport Model, aiming to simulate the filtration and reabsorption function of the kidneys using a semi-permeable membrane. For this experiment, I needed a dialysis tubing of 10 to 15 centimeters pre-soaked in water, string or rubber bands, starch solution, glucose solution, iodine solution, Benedict's reagent, 2 beakers, 2 test tubes, and a test tube rack. Then I needed pipettes or droppers, hot water bath, and distilled water, then a marker and data sheet. Unfortunately, we weren't able to purchase any dialysis tubing from the local pharmacies and medical supply stores, and the hospitals or dialysis centers here didn't have any to sell. As a result, I improvised and used vegetable cling film instead. This is a viable alternative due to the presence of small pores in the plastic which allow molecules between 0.01 to 10 nanometers in size to pass through. However, this change should be noted as it significantly affects the expected results. To start, I had to prepare a mix of equal parts starch solution and glucose solution in a beaker. This was then transferred into the cling film and tied tightly to seal. The outside should be rinsed with clear water and checked for any leaks. There should be no sign of leaking or breakage. Then, the bag is lowered into a beaker of some iodine solution. Here, I tried to make sure the bag was not submerged all the way in case some may seep from the top and skew the results. Over time, the starch molecules in the bag turned dark. This is due to the formation of a charge transfer complex, absorbing a different wavelength of light and causing the dark color. This reaction indicates that polyiodide ions crossed the semi-permeable barrier and created a complex with the starch molecules. Next, we have to test if any glucose molecules from inside the bag crossed into the outside solution of iodine. To do this, I had to take a small amount of the outside solution and put it into a test tube. Then, I added an equal amount of Benedict's solution. The initial green color does not yet indicate a reaction, only that the yellow and blue pigments from each substance mix together. The reaction will only take place when the solution is brought to very high temperatures. This would only take around 30 to 60 seconds when placed in a bath of boiling water. I made a replica to observe and compare the results after. Typically, Benedict's solution will show different levels of color change depending on the concentration of glucose. If glucose has leaked into the iodine solution, this would result in a green, yellow, orange, or red color change. The solution remains blue. This indicates that no glucose molecules managed to cross the barrier into the outside solution. The iodine molecules had decomposed due to high heat, turning clear or white color, with no glucose molecules to react with the Benedict solution, leaving only blue. After thorough rinsing, the solution inside the bag was removed and poured into a beaker. This was then sampled and mixed with Benedict solution into a test tube, similar to the previous ones. After heating, the solution had turned into a deep color, meaning the solution inside the bag had very high levels of glucose. The experiment demonstrated how a semi-permeable membrane controls the movement of substances based on molecular size and membrane permeability. In this model, iodine molecules were able to cross the membrane and react with the starch solution inside the bag, causing the contents to turn dark blue-black. This indicates that iodine particles were small enough to pass through the membrane pores. However, glucose molecules did not appear in the outside solution because the Benedict's test remained blue after heating. This suggests that the cling film used as a substitute membrane was less permeable than actual dialysis tubing, preventing glucose diffusion. Starch was unable to cross the membrane because starch molecules are large polysaccharides composed of many glucose units. Their size prevented them from fitting through the small pores of the membrane. This reflects glomerular filtration in the kidneys, where only small molecules such as water, salts, glucose, and urea normally pass through the filtration membrane, while larger molecule substances like proteins and blood cells are retained in the bloodstream. If proteins or blood cells had been placed inside the tubing, they would also likely remain inside because they are much larger than glucose or iodine molecules. In normal kidney function, these substances are not filtered into urine because the glomerular membrane acts as a selective barrier. Similarly, glucose is normally absent or only present in trace amounts in urine because it is filtered first and then reabsorbed back into the blood by the kidney tubules. Significant glucose in urine may indicate impaired reabsorption, such as in diabetes mellitus. Overall, the experiment showed that diffusion depends on both concentration gradients and membrane properties. Molecules naturally move from areas of higher concentration to lower concentration, but whether they can cross depends on their size and the permeability of the membrane. The cling film membrane allowed iodine diffusion but restricted glucose movement, demonstrating how membrane structure can greatly influence transport outcomes. In conclusion, the membrane transport model successfully demonstrated selective permeability and diffusion across a semi-permeable membrane. Iodine molecules crossed the membrane and reacted with starch, while glucose did not diffuse into the outside solution due to the limited permeability of the cling film membrane. The experiment simulated how the kidneys filter small substances while retaining larger molecules such as protein and blood cells. Overall, the results showed that membrane transport is influenced by both molecular size and membrane. Overall, the results showed that membrane transport is influenced by both molecular size and membrane properties.